Very good morning, students. I hope you all are keeping well. After Diwali vacation, today we are going to continue with the second unit test portion with lesson number 11 of subject Science. Lesson number 11 in science subject is cell structure and microorganisms. Now, let us understand the meaning of cell and how they are formed one by understanding different examples. Now, what is the name of minute component of which the body of a living organism is made? Means you know that all living things we have particular structure, okay, body, different organs, different parts of the body are there and they are made up of particular things. Now what are those minute components? What are those things of which this body of a living organism is made? We have learned that cellular organization is the primary characteristic of all living organisms. Means what? What I said, that cellular organization means what? The cluster, the group of particular thing, correct? Organization, when many things comes together, it's called organization. So cellular organization is what? That the many cells comes together, correct? Cellular organization where that many cells have came together is the primary characteristics of all living organisms. Means in every living organisms, different cells have come together and formed one organization, one structure, one part of the body, correct? Then what do you mean by cell? Okay, let's start with the definition of cell. Cell is nothing but the fundamental, structural and functional unit of living organisms. Definition of cell is what? The cell is the fundamental, structural and functional unit of living organisms. Means if you go on dividing or if you find out the smallest component of a living body, then that smallest part, that smallest component is called as a cell. It is fundamental, means it's a basic, correct? Means if one cell is of particular type, all other cells will be of same type. Okay, for example, if you take example of a heart, or if you take example of kidney, whatever cells they are made up of, correct? All cells will be same in the organ heart or in a kidney. Means that particular organ is made up of fundamental cells, means each and every cell will be of same type. Then structural and functional unit of living organisms, means each cell has a particular structure, has a particular function in that body. Okay, this is the meaning of definition. Okay, shall we repeat? The cell is, along with me all of you repeat, the cell is the fundamental, structural and functional unit of living organisms. Now we'll take one example, then you will understand it in a proper way, correct? In a figure or on a screen you can see one flow chart is been shown. Very first it is given a letter. Okay, letters. Which are the letters? A, B, C, D, these letters. Okay. Two or more letters comes together and form one word, correct? Using the letters, words are formed. Many words comes together and sentences are formed. Then different sentences comes together, paragraphs are formed. Many paragraphs comes together and one chapter is been formed, correct? And many chapters comes together, compiled together, combined together and one book is been formed, correct? So can you see the flow chart? Flow chart means what? In one direction it's flowing down, or its direction is from up to down. There is a sequence, Okay, letter to words, word to sentence, sentence to paragraphs, paragraphs to chapters, and chapters to book. Okay, so this is example of letter or a book. That what we say, that the structural organization of book, this we can call it as a structural organization, means how the book is been formed. Similarly, there are organizational level in living organisms too. Okay, means if any one organ has to be formed, then there is a series of particular flow charts. Correct? For example, let us see how the organs are formed. On the screen you can see the figure. Okay, one, initially you can see the cell. Many cells comes together and tissue is being formed. Correct? Many tissues comes together, different types of tissues comes together, and one organ is formed. Okay, and then many organs comes together and one organ system is being formed. Okay, this is the example of one digestive system they have shown. Same way each system in a body is being formed. Okay, respiratory system, let it be digestive system, let it be excretory system. Each system is made up of a single cell. Are you getting it, children? What we have said? We have said that structural organization, cellular organization. Okay, so in this figure, the organ, there are the organizational level in our living organizations, living organisms. These are cells, tissues, organs, organ systems, organisms. Okay, cell form the basic of the structure and functions of all living organisms. It's only with the help of cells that living organisms carry out all their different life processes. How these life processes takes place, that we will see one by one. First, let us understand how these cells are formed and how we can study with the what instrument we have to use to study the structure of cells. Structure and microorganisms part 2. Having learned the definition and meaning of a cell, now let us try to understand how the concept of cell came into existence and how this study began. In 1665, a scientist whose name was Robert Hooke. Okay, as you can see on the screen, one picture is been shown, and his name is he is the scientist. His name was Robert Hooke. Okay, so what he did, he observed a thin section of a cork under a microscope. Cork means what? You can say the cap of any bottle, rubber cap. Nowadays we are having metallic caps, but in ancient days that cork, that cap of the bottle was made up of wood or rubber. Correct? That material, that is cap of that bottle, is called as a cork. He observed a thin section of a cork under a microscope. So microscope, what is microscope? Microscope is an instrument which can show you the expanded image of the object, any object which you want to observe. Correct? In the picture you can see on the right hand side, they have shown the structure, or you can say this random, means initially how these microscopes were looking like. These are the microscopes which were used in ancient days. Afterward they are developed, and nowadays very much advanced instruments are available. Correct? But the Robert Hooke was the scientist. What he did, he took a thin section, he cut it, that cork, into thin section, and he observed it under a microscope. So what he saw hexagonal compartments. Hexagonal means what? The figure which have in maths we have studied, figure which have 6 sides. Okay, like a honeybee. Honeybee makes its house, that is of hexagonal shape. So he saw the hexagonal compartment like those of beehive. He named those compartments as cells. Means each part, that hexagonal part, what he named it as a cells, and this term is derived from the Latin word cella, which means the small rooms. Okay, now in 1838, two scientists M.J. Sheldon and Theodore Schwann formulated a theory about cellular structure which stated all the living organisms are made up of cells, and the cell is the fundamental component of living organisms. Means initially the word cell was given by Robert Hooke. He was studying about that different thing. Cork section, he named it as a cell. But afterward, M.J. Sheldon and Theodore Schwann, they formulated one theory that each living organism is made up of cell. All the living organisms are made up of cells, and the cell is the fundamental component of living organisms. In 1885, Rudolf Virchow stated that all the cells are formed from pre-existing cells. Okay, so one cell, if one cell is existing, that cell must have formed with the help of previous same kind of cell. Means the new formation of new cell. For example, formation, if you take example of a flower, okay, rose flower. This rose flower, whatever cells it's having, it's formed with the same type of cells, originated from the same type of cells. So this was in short they have given us history to understand that how these scientists have started the studies. So in next part of our videos we will study how to observe these cells. Means they are so small, we are saying the smallest part of the organism, smallest part of the living organism. Then how to observe that, how to study that? So we have a instrument that is called as a microscope. Let us see the structure and function of that microscope in our next video. Lesson number 11, Cell Structure and Microorganisms, Part 3. Measurement and observation of cells. Now, in 1673, the scientist whose name was Anton van Leeuwenhoek, he assembled various lenses together to construct a microscope. He was the first one to observe live bacterial cells with the naked eyes. Correct? So, till 16th century, the scientists were aware of the cells, concept of the cell. But how those cells looks like, what are the different organelles present inside the cell, they were not knowing it. Okay, so the first scientist Leeuwenhoek, he made the microscope. What is a microscope? Is the combination, is the assembly of many lenses, okay, different capacity lenses, they are fixed together. And what it happened, the things which are very minute, very small, which you can't see with the naked eyes, means with our normal vision we can't see anything, that we can observe using a microscope. What microscope do? It expands the images. Okay, it expands the images to 100 times, 1000 times. Many means the lenses are of different capacity. Based on the requirement, we have to use the lenses and microscopes. Okay, so in our book they have given one diagram. So it's a basic, that is compound microscope. In this one, as you can see in the figure, okay, upper part, this part is here is upper one. Okay, this is called as eyepiece or ocular. You have to observe from wherever I have drawn this line, this is eyepiece. Okay, from here you have to observe the things. Within this rod, this tube rod, as I am marking with the red lines, okay, you can see that this rod, tube like structure, lenses are fixed inside that one. Okay, and these are the objective lenses. You can see this one that rotates, you can rotate it with, you can see there are three objective lenses are there, 1, 2 and down back side it is 3. So whichever lens and these all lenses are of different capacity. Okay, for example, if I want to observe the parts of flower, now flower parts, as you know, I can see the flower parts with my eyes, they are not very minute. So I want the lens of little bit low capacity, means even the low capacity lens can expand the flower. So I will use the objective lens for this observing parts of flower. Suppose if I want to observe the cell of the leaves, okay, green leaf if I take from any plant, and if I want to observe the cell, then that cell is very minute part, very small part. I require high power lens because it has to focus or it has to expand the object more. So these are the different objective lenses. According to our requirement, that much power lens we have to use. These you can see coarse force focus knob and fine focus knob is there. These are for what? For fine adjustment, means whenever you rotate these knobs, we have to rotate it, okay, front back movement you have to do. Then what happen, this tube like structure moves up and down, little bit movement takes place, and the object which you want to see gets focused. The way, for example, if I give you example, when you click the pics with the mobile phone, okay, so what you do, or any camera, when you click the pics, first you focus, okay, by tapping your mobile screen you focus the part which you want to highlight. That same way the microscopes also have the fine adjustment. You have to do these focus knobs, you have to adjust these focus knobs, correct? And this is stage, you can see the stage. On this we have to keep the slide which you want to observe, okay, there you have to keep one glass slide, and on that glass slide the observing material we have to keep. Downside you can see they have shown the illuminator. Illumination means what? Making thing bright. In short, it's a mirror like structure or lens, it's there. It is there to increase or decrease the intensity of light, means some bacteria or you can say some small animals which you require a dark light to observe, sometimes you require a bright light, so that light adjustment is done by the illuminator. And the downmost part is the base. Base means is the you can see the U-shaped structure, it's a base. So this is the basic structure of the microscope, correct? Using this microscope, it's called as a compound microscope. It's a very basic microscope which we use in a school level or college level. If you go on to do some research work or in a laboratories, the microscopes with a higher capacity, with a higher capacity lenses, and nowadays many advanced microscopes are also there which are directly connected to a computer screens, okay. You just have to, on microscope you have to click, you have to focus, and on computer on screen you can get the picture also, means there is no need to see through these tubes, okay. So basic structure of compound microscope, it is given in our textbook. Lesson number 11, Cell Structure and Microorganisms, Part 4. In Part 3, we have seen the structure of microscope, compound microscope rather, okay. Now we are going to continue with the part how to measure the size of cells, correct? Now as we have seen that cells are extremely minute in size and we cannot see the cells with the naked eye, correct? And therefore we are using an instrument, microscope, okay. Microscope expands the image of the object, okay, and it's clearly visible to us. So object on a glass is magnified, you can say expand or magnified, okay. lenses are there which magnify the image by many times, which magnify the object many times by the lens of the microscope. Now, how to measure the size of cells then? As we are saying, the size of cell is very small. We cannot observe it through naked eye. Then, how to measure the size of the cells? Okay, so scientists have discovered certain units. Now, on the screen you can see, first in a green band I have written the unit 1 centimeter. You all are familiar with the unit 1 centimeter and millimeters. Correct? Till millimeters we are aware that the smallest unit till now which we know is the millimeter or milliliter. Now we are going to learn the units which are smaller than millimeters. 1 millimeter distance is there and many cells are smaller in size. Correct? So 1 centimeter is equal to 10 millimeters. Next, 1 millimeter distance. Now 1 millimeter, you know, you can take out your scale and 1 millimeter distance you can see. Okay, so that 1 millimeter distance is equal to 1000 micrometers. Correct? Means that small 1 millimeter distance, if you divide it into 1000 parts, so you can imagine, okay, that 1 millimeter's 1000 part means how much small it will be. Okay, so that 1 millimeter is equal to 1000 micrometers. In short form, the micrometers are also called as microns. Okay, now what are the microns? Yes, still microns or micrometer is further divided. 1 micrometer means the 1000 part of 1 millimeter is equal to 1000 nanometer. So the smallest unit till now we know is what? Nanometer. Means you can just imagine how much small must be the size of cell or size of any object. Correct? And that is why we require the microscope to measure the small distances. Okay, so let us repeat it again. 1 centimeter is equal to 10 millimeter. 1 millimeter is equal to 1000 micrometers. Micrometers are also called as microns. And 1 micrometer is equal to 1000 nanometers. So these are two new units which we have learned today, that micrometers and nanometers. These units are used to measure the size of cells or very minute particles or very minute organisms. Okay, so one example in our book they have given, that is onion. Okay, so that example, let us see what they have explained. Lesson number 11, Cell Structure and Microorganisms, Part 5. In our textbook, it's given one method to observe the cells of peel of the onion cells, or onion cells we have to observe under a microscope. What we have to do in this procedure? You have to take a small piece of onion and carefully separate the skin from its concave surface with the help of forceps. Okay, so if you see the onion part, okay, normally you don't have to take a thick part. This is a onion. So if you see this shiny portion is there, okay, it's nothing but the thin covering. Onion is covered with the colorless segment, or colorless, you can say, membrane. Okay, it's covered with the membrane. That membrane we have to peel it up and you have to separate it out. Keep it on a glass slide. Okay, this is the segment or membrane is there. From that segment, a small part we have to take. Correct? So then keep the membrane on slide. One or slide we have to take, and on the slide we have to keep the membrane. Slide is what? A small rectangular piece of the glass. Okay, it's a transparent glass on which this membrane has to be kept. It is mixed with, or we have to put a drop of some dilute solution of iodine or eosin. Okay, now iodine or eosin. What are these iodine? Iodine and eosin, EOSIN. These are some coloring agent or dyes, you can say. Okay, eosin, iodine. Anyone we have to use dilute solution. Dilute means what? Iodine you might have seen in our first aid box. Sometime it's present, brown colored liquid is there. At that dilute solution of the iodine. Dilution means what? Strong concentrated solution if you mix it with the water. Suppose if you take 10 milliliters of water and put 1 drop of iodine, so that will be a dilute solution of iodine. Eosin also is the stain or coloring agent, you can say. It's a pink or reddish pink in color, and that dilute solution we have to put on the membrane. So what will happen? This is the colorless, membrane is colorless. It will give the slight pink or slight brown color to membrane, and because of that it's easy to observe. As you can see in this structure, this one, okay, you can see the strip like membranes, strip like cells are there. Okay, so what you have to do? Take a slide. On a slide you have to put the membrane. On membrane you have to put the iodine or eosin solution diluted. Okay, 1 drop. Solution means you don't have to pour 10 ml. Only 1 drop we have to put, and that slide we have to observe under a microscope. 10x objective. Okay, we have learned that there are different powers. Objective lenses are of different powers. So 10x. 10x means what? 10x power lens we have to take, means 10 times stronger, 10 times more. It will give you the size 10 times more size. Okay, so 10x objective we have to select, and before observing we have to put one small cover slip on that slide. Cover slip is a very thin and very slender thin slide, thin piece of the glass which you have to put over the membrane and observe under a microscope, and we will be able to see the structure which as they have shown in D part. Okay, the cells like rectangular cells, circular cells. This is the onion cell. So this kind of structure you can see. If you take any other different part of plants like leaves, bark, root tips, then different types of cells we can observe. Okay, so it's basic method to observe the cells of the living thing. Now in our book they have shown different types of cell, various cells of various size. Okay, cells are of different shapes, different sizes, depending on the organ, depending on the what you say, any organization of which it is a part. Okay, so let us study the cell size and different shapes of the cells. Lesson number 11, Cell Structure and Microorganisms, Part 4. In Part 3, we have seen the structure of microscope, compound microscope rather. Okay, now we are going to continue with the part how to measure the size of cells. Correct? Now, as we have seen that cells are extremely minute in size and we cannot see the cells with the naked eye. Correct? And therefore, we are using an instrument, microscope. Okay, microscope expands the image, expand the image of the object. Okay, and it's clearly visible to us. So, object on a glass is magnified. You can say expand or magnified. Okay, lenses are there which magnify the image by many times, which magnify the object many times by the lens of the microscope. Now, how to measure the size of cells then? As we are saying, the size of cell is very small. We cannot observe it through naked eye. Then how to measure the size of the cells. Okay, so scientists have discovered certain units. Now on the screen you can see, first in a green band I have written the unit 1 centimeter. You all are familiar with the unit 1 centimeter and millimeters. Correct? Till millimeters we are aware that the smallest unit till now which we know is the millimeter or milliliter. Now we are going to learn the units which are smaller than millimeters. 1 millimeter distance is there and many cells are smaller in size. Correct? So 1 centimeter is equal to 10 millimeters. Next, 1 millimeter distance. Now 1 millimeter, you know, you can take out your scale and 1 millimeter distance you can see. Okay, so that 1 millimeter distance is equal to 1000 micrometers. Correct? Means that small 1 millimeter distance, if you divide it into 1000 parts, so you can imagine, okay, that 1 millimeter's 1000 part means how much small it will be. Okay, so that 1 millimeter is equal to 1000 micrometers. Okay, in short form, the micrometers are also called as microns. Okay, now what are the microns? Yes, still microns or micrometer is further divided. 1 micrometer means the 1000 part of 1 millimeter is equal to 1000 nanometer. So the smallest unit till now we know is what? Nanometer. Means you can just imagine how much small must be the size of cell or size of any object. Correct? And that is why we require the microscope to measure the small distances. Okay, so let us repeat it again. 1 centimeter is equal to 10 millimeter. 1 millimeter is equal to 1000 micrometers. Micrometers are also called as microns. And 1 micrometer is equal to 1000 nanometers. So these are two new units which we have learned today, that micrometers and nanometers. These units are used to measure the size of cells or very minute particles or very minute organisms. Okay, so one example in our book they have given, that is onion. Okay, so that example, let us see what they have explained. Lesson number 11, Cell Structure and Microorganisms, Part 6. Cells are of many different shapes. For example, some are circular, some are rod shaped, some are column like shaped. Okay, you can call it as columnar shape, spiral, oval, rectangular. Many types of shapes are there for different cells. Depend on which organ, in which organ it is present. Based on that one, the shapes are also different. Each cell contains various components for carrying out the life processes of the living organism, and these components are called as organelles. Means till now what we have seen, that cells are very minute in size. Cells you cannot see with the naked eyes, and inside the cell also again different components are there, and they are called as organelles. Okay, I repeat, just children pay attention over here. Each cell contains various components. Means cell is very small in size, it's very minute in size. Inside the cell again some components, some things are present. Okay, why they are present? For carrying out the life processes of living organism. What are the life processes? Okay, our respiration, breathing, okay, purification of blood, excretion, digestion, these are all life processes. Without them we cannot survive. Even one process is been disturbed, our full body cycle gets disturbed. So these cells contains components, they are called as organelles. Correct? So inside the cell again small components are present, that components are called as organelles, and these organelles can be studied with the help of electron microscope. Now we have learned the structure of compound microscope. Whichever microscope we have learned, its name was compound microscope. But with the compound microscope, we can only visualize, or we can only see the structure, only cell size and color and all that. But inside the cell again some organelles are present. Okay, to see that organelles we need a different developed type of microscope, and that is called as the electron microscope. In short, in simpler language, it's a powerful microscope. Okay, it can magnify the images up to 2 billion times of their actual size. 2 billion times more it expands and show you the things. In our book they have shown us the different structure or various cells. Okay, you can see in the picture amoeba, spirogyra is there. Let us highlight them. Okay, this is amoeba. Amoeba is a unicellular organism. It's a very minute organism. Normally they are present in polluted water, or you can say the ponds, streams, there. Okay, so these are unicellular, means their body contain only one cell, correct. Then they have shown us the red blood cells. In our blood is of red color, as you know, but that red color to the blood has received because of these red blood cells. How they look like, now you can see the structural difference between these two, amoeba and red blood cells. Okay, their shape is different, their color is different, of course their function is also different. Then fat cell they have shown, egg cell, bone cell, nerve cell, muscle cells, sperm, correct. So these all types of cells are present in living organisms, and each and every cell has the different color, has the different size, has the different shape, and has different function also. Okay, as we have seen, the definition of the cell is what? Cell is the fundamental structural and functional unit of living organism. Okay, so each organ have the fundamental structural and function, means each cell has the basic shape, basic structure. Now shape and structure is different, don't get confused, and have different function. So there is great variation in the shape of cells. These Standard 7, Subject Science, Lesson number 11, Cell Structure and Microorganisms, Part 7. Till now, we have learned what do you mean by the word cell, meaning of cell, then the instruments which are used to measure the cell size, and different types of cells. Today, we are going to learn the main two types of cells, that is plant cells and animal cells. There are two main types of cells, plant cells and animal cells. Plant cells and animal cells consist of various types of membrane bound cell organelles. What do you mean by cell organelles? Yes, the way our human body or any animal body have the different organs, correct? Inside our body there are organs, okay, like heart, kidney, lungs. Different organs are present inside the body. Same way, there are small organelles are present inside the cells, and they are called as organelles. Okay, it's that word organelles word is similar to word organs. Okay, same the way we have the different organs, cells have different organelles. Plants and animal cells have definite shape, correct. We are going to learn two main types of cells, that is plant cells and animal cells, because there is difference between structure of plant cell and structure of animal cell. Okay, so basically what is the difference? What is the structure of each cell? Some or other as the animals or as the plant changes, some or more little bit differences will be there, but the basic structure will be same. Plant cells have definite shape due to the presence of cell wall around them. Besides, unlike animal cells, plant cells contain single large vacuole, and all these cells are known as eukaryotic cells. To understand this, let us look at the figures which are shown in the slide. On left hand side, you can see is the diagram of plant cell, and on right hand side we can see is a diagram of animal cell. Okay, so we can see there is a difference between the structure, shape of the plant cell and animal cells. Okay, now these plant and animal cells have similar differences, several differences and similarities. Okay, means some differences will be there. Some of the organelles are present in plant cells which we will not find in animal cells. Okay, there will be the difference between plant cell and animal cells, as well as there are particular similarities. For example, animal cells do not have a cell wall or chloroplast. Okay, so that you will understand once you will learn the different organelles and what are the different functions in short. Okay, in 7th standard we don't have to learn these organelles in detail, but it's the introductory part. We are supposed to learn about the structure of the plant cell and animal cells. You can see the figures, or you can see the diagram of plant cell. Okay, it's a hexagonal shape, and that shape you can see it's a definite shape. It's not a irregular shape. Correct? Whereas if you see the animal shape, though it's a rectangular shape, but there are certain notches, there are particular curve at particular sides. Okay, so you can visually see, or visually you can make a mark out the difference between the shape of the plant cell and animal cell. Okay, we can see in diagram they have labeled different organelles or different pink colored, yellow colored parts inside the cell, and what all these parts we call them as a organelles. Okay, these all parts we call them as a organelles. Now first we will just go through the some basic organelles or basic part of the cell, then again we will learn these organelles or these main parts of the cell with the help of diagrams. So the cell has four main parts: the cell wall, cell membrane, cytoplasm and cell organelles. Let us study them in detail one by one.